So at the top left here, go ahead and draw a circle. Okay. What we're going to draw here is Staphylococcus aureus, the bacteria that developed this resistance. I want you to think of it like a ball. It's a three-dimensional ball. Our diagram is going to be kind of a cross-section. We're going to cut it in half so we can see what's inside, but also what's on the outside. So this type of bacteria, it's got actually two barriers that protect its internal components. On the outside, it's got this thing called a microcapsule. A microcapsule. It's like the first line of defense protecting the inside of this bacteria. But then it also has this little internal wall that's called a cell wall. Now, they share the function of protecting the inside, but they have slightly different compositions and made of slightly different things. But they protect the most important thing inside the bacteria, which is its DNA. And when you look inside of like a human cell, not a bacteria, normally the DNA is a lot more organized and it's inside something called a nucleus. In a bacteria, there's no nucleus. It's actually just kind of like this big clump mess of DNA. And so they refer to this as its main DNA, which controls its traits. But then what's also kind of weird about this type of bacteria is they have these little small pieces of DNA that have kind of pinched off the main piece. And we call this a plasmid. And they're very small pieces of DNA. They're usually circular. And they contain a small amount of DNA that actually controls some specific traits. Okay. Now, there are lots of other things contained inside the bacteria, but these are the components that we need for our story. All right. So on the other side, we're going to start with this guy named Alexander Fleming. And Alexander Fleming was this scientist who was studying Staphylococcus aureus particularly in the 1920s. Now, just a quick context. In the 1920s, that was 2 years after historically one of the worst outbreaks of the flu. In 1918, there was the influenza pandemic that resulted in the death of hundreds of thousands of people. That influenza is like very similar to the flu that people catch today. It was actually a strain that spread very quickly. The reason that's important is because in the 1920s, that kind of led to a huge push to study disease. Because not too long ago, bacteria like this one, it causes pneumonia, and also the flu, which causes, as in that case, influenza, and a lot of the undesirable symptoms. People didn't really understand it. And so researchers spent a lot of time, a lot of money was put into trying to understand disease for both prevention and also treatment. And so this was one example of that. And because we didn't really know a whole lot, his research mainly consisted of taking what's known as a petri dish. So it's usually a plastic or a glass plate. And you would actually spread bacteria all over it. And watch them grow into these little, what they call colonies. Now this is kind of zoomed out. You can actually see this without a microscope when you grow bacteria like this. Each dot there that you see is not one bacteria. It's actually on the order of probably closer to 1 million bacteria within that one dot. We can think of that as like a population of Staphylococcus aureus inside of each one of those little dots. Called a colony is what they call it. So he would do this over and over and over again and would try and change things about its environment to see what would happen. But then when he pulled out a few plates this one time, he noticed that like on this one portion of the plate, no bacteria were growing. And this bacteria normally just kind of spread all over the plate. There was never an inch that was like not taken up by at least a few colonies. And so he pulled this out, and there was just like this hole. And so it begs the question, well, what the heck happened? Right? Why was there no bacteria? And the weird thing about this was that when he looked at this plate, he started to kind of take samples off of that little region that I put a question mark around. And he actually realized that he had contaminated his plates with a species of mold. And so mold, like you might see like on a piece of cheese that's old or like a piece of bread, it's a fungus. And what he saw was that he realized a species of mold called penicillin. The mold was actually called penicillin, had contaminated his plate or his petri dish. And that could have been for a variety of things, like either he was eating his lunch near this set of plates. It could have been that his lab wasn't properly cleaned, but it was by accident that he realized this. Okay, so he realizes that it's penicillin. He realizes that there's no bacteria growing. And so the question was, what was this mold doing to prevent growth of Staphylococcus aureus? Right? Because if we could figure that out, then maybe we could channel whatever this fungus is doing to make it useful. Okay. So after a couple years, he actually realized that the mold produced a substance that interrupted the assembly of new cell wall. Okay. So, what this means, going back to our diagram there at the top left, I told you there were two boundaries. There's a microcapsule in a cell wall. Now, when a bacteria grows, like on this plate, into hundreds of thousands of bacteria, it uses a process called binary fission, is what they call it. Binary fission. And all that happens here is that all of the DNA and plasmid DNA, it replicates. They also make more cell wall and more microcapsule, and so they can make a complete copy of the bacteria with a full set of the DNA and plasmid. But in order to do that, you need more cell wall, you need another copy of the DNA. Otherwise you can't divide, you can't grow. So down here, I mentioned that mold produces a substance that actually interrupts cell wall, new cell wall, from being assembled into those new replicated bacteria. So if that can't happen, the bacteria can't replicate, they can't grow. So this prevents S. aureus from growing. They can't divide in the presence of that substance. Okay. I'll pause for a few moments. Make sure you catch up if you're a little bit behind. Are there any questions so far? Penicillin is a mold, produces a substance, interrupts cell wall assembly, which prevents the growth of Staphylococcus aureus. And this was the first time they'd ever found a substance that could interrupt that process. And so what they did, they isolated, meaning they took it out, this substance, and called it an antibiotic with the specific name of penicillin. Because it was an antibiotic, which means it kills bacteria, derived from the species of mold called penicillin. Okay. Yeah, so the question she asked was, are there different types of mold? Yes. What might be another type of mold that we saw in this story? Remember the other drug that was introduced called methicillin? Right? That was actually derived from a different species of mold as well. So the short answer to your question is yes, there's actually tons of types of mold that each produce kind of different forms of antibiotics. Okay. Good question. So, we're almost there. So we have this substance. And let's think about how traits change over time, right? So the colonies on that plate we can think of as like a population. Right? little bit different looking than a population of animals or humans, but it's a population nonetheless. What is our trait of interest? What is our trait of interest that might be relevant to bacteria being resistant? What do you think, baby?
Bacteria?
What traits in the bacteria might be relevant to a bacteria developing resistance?
It was able to, I don't know the word, but it was able to fight like penicillin, right? So, I don't know how to explain it.
Okay. Does everybody see what I'm asking? Right? We went over a couple of different traits of this bacteria, and in the media framework, there's a trait of interest that's relevant to what's changing. So, Annika, what thought do you have here? Yeah. So their cell wall is a trait of the bacteria that if variations in the cell wall come about, might that produce some resistance? Because that's the part that the penicillin affects, is the cell wall. And that's actually what was observed, was that random mutations occur in the DNA of Staphylococcus aureus, which create variations in the cell wall. So some members of the population, their cell walls, they actually look a little bit different than the cell walls of other members of the population. You might see where this is going. There are some variations. This is kind of our last big point. There are some variations in the cell wall which are resistant to the effects of penicillin. So penicillin, again, that substance, it interrupts the assembly of the cell wall. There's some variations in the cell wall that actually penicillin can affect. So even in the presence of penicillin, Staphylococcus aureus can still divide and therefore is resistant to the drug. And based on what we know about adaptation, what's going to happen to the frequency of that variation in the population? Is it going to become higher or lower frequency? Higher, right? Over time, the ones that survive that have these variations are able to divide or reproduce, and therefore that variation in cell wall is going to become more and more and more common. Okay. So, when we look at this story, we started with this random dude, Alexander Fleming, who by accident discovered penicillin in the mold, which then led to the isolation of a substance that's our first antibiotic. But then, like any other living organism, mutations occurred that affected the cell wall, which led to those variations becoming more common in the presence of penicillin, because they could survive. There's two kind of lingering questions that before I pose those to the class, are there any clarifying questions or curiosities that come up after listening to that? This is also recorded too, all of the notes in case you missed it. Okay. The first question that I think comes up here is, in the story, methicillin was introduced after penicillin resistance became common, right? Do you think methicillin affects the assembly of the cell wall, or do you think methicillin affects something else? Did everybody see what I'm asking? Affects the cell wall. The effect of the cell wall not going to work. Right? Variations, but methicillin and do different things. Some types of antibiotics is why fungus. Why is fungus producing a substance that actually... I want you to turn to your neighbor real quick. Why would that be useful for a fungus?